Okay. I think we are going live, right? Okay.
Okay.
Great. Okay. Hello, everybody. I'm happy to welcome you in this session number 6. So the topic of this session is environmental and self-motion-based navigation. And as in the previous session, we will have 4 speakers. And our first speaker is already on stage. His name is Thackery from Georgia Institute of Technology. And his talk will address influences of environmental barriers on human entorhinal cortex and cognition. And really looking forward hearing your talk, Thackery. So please go ahead. The floor is yours.
Okay. Thank you very much for having me. It's a great honor and pleasure to be here and speak to you. I apologize in advance for my froggy throat. I'm a bit under the weather, but I do have some water with me. To sort of introduce myself first, I'm an assistant professor. And so maybe not all of you are aware of the sort of work we do in our lab. So more broadly and generally, what we're interested in my group is the mechanisms that enable us to form memories for different experiences that we have in our environment, and our ability to leverage memories for those experiences in service of planning and of goal-directed behavior. And I've been focused for many years now on neuroscientific questions surrounding the hippocampus. So we've conducted a number of studies looking at how the hippocampus enables retrieval and even the mental simulation of different routes through an environment, despite shared contexts and potential interference between memories. So I've highlighted an example of this here in yellow. And what we've become really interested in recently is the profound behavioral variability that we see in these tasks. So I plotted an example of this on the bottom left here. This is a paper we actually have under review right now, but using some paradigms that we've used for a long time now, so the map may look familiar to you. We were struck by the great variability in the degree to which different people acquire configural knowledge of an environment when they're forced to learn it from these very strict route-based learning scenarios. So you can see from this plot of pointing errors in the environment from a start to a finish point that some participants really, really struggle with this, even though they may do the navigation task just fine as probed. So this led us to become really interested in whether we can relate this type of variability in people's understanding of the environment to neurocognitive traits and perhaps how they process the features of the environment that they've tried to learn. So today I'm going to give a rapid overview of some of the work we've been doing to understand these individual differences in spatial learning via virtual reality manipulations of environmental features. And I wanted to highlight, you can kind of flip this theory-oriented question on its head and take an applied angle and ask, can we manipulate virtual environments to improve spatial learning? And perhaps can we do this in a manner that's tailored to individual differences? And one thing that's important if you take this applied angle is that any learning benefits you might get from manipulating virtual environments, you want them to transfer to naturalistic versions of the environment, or perhaps even the real-world variant of the environment that you're simulating. So you're going to see this pop up in the experiments that I tell you about today, and kind of note that that is a feature of what we find. So what I'll focus on are actually 2 spiritually connected research directions, one cognitive and one neural, on how people perceive their interactions with barriers in virtual environments. And barriers have become very interesting to me. A lot of research from many of you here in this audience, in psychology and neuroscience and geography, a lot of research from your groups has shown that environmental barriers can disrupt our representations of space. They can distort our estimates of distances and our sense of angles between different features of the environment. And so the interesting question for us is, why is this? In our lab, we've operationalized these barriers as things that can cause disruptions of continuous movement. And this can happen through disrupting visibility as well as the actual navigation affordances in the space. And my amazing postdoc, Shiliang He, who deserves the credit for the work that I'm showing you today, he previously conducted a study using virtual reality to understand how affordances affect spatial learning. And what he did was he manipulated the penetrability of barriers in virtual environments. So participants could basically drive through buildings during learning. And what he found is that at test, if he took away this penetrability power and probed participants' spatial memory, they exhibited improved straight line knowledge of the relationships between locations in the environment. But interestingly, not so much their wayfinding ability. So it's almost like this manipulation, through this manipulation, they lost something about the navigable geometry of the space. So this was interesting to us, and we began to wonder what role visibility plays in cognitive map formation. So can we improve wayfinding performance, perhaps through perceptual manipulations of the environment? And so we had two hypotheses, which I imagine most of you would find intuitive. So one of these is that reduced visibility hinders our ability to directly perceive spatial relationships. And because of this, we have to infer spatial relationships over the course of an encoding event. And we know this is an error-prone process. And our other hypothesis is that good navigators on these sort of tasks may not necessarily be individuals who have superior long-term memory abilities, although certainly that would help. But maybe they're also individuals who simply use the spatial cues that are available in a more flexible and adaptive way. So if this is true, and we have a manipulation of barrier visibility, this would open up more visual cues to individuals. And what this would mean is that that manipulation would be more effective in individuals who are better at leveraging these additional cues to understand the layout of the environment. So to go after these hypotheses, we use the same general paradigm that Shiliang used in his navigational affordances experiment, but instead of manipulating whether barriers are penetrable or not, we granted participants X-ray vision to reduce this sort of visibility hindrance component. And the idea here is that if we preserve the navigational affordances in the environment, then participants will still learn, perhaps implicitly to some extent, more about the navigable geometric structure of the environment in addition to the sort of visual layout. And this may better transfer to wayfinding performance on subsequent probes of their ability. So in this task, what we had was a relatively small environment with 9 buildings, and on each building there were 4 different storefronts. And over the course of 2 training sessions, participants would explore the environment, and they'd gradually learn where 9 target storefronts were. And the thing to highlight is this task was actually quite difficult despite its small size. So there's no global landmark, and you'll note the buildings are all rotated relative to one another. So there's a relatively complex relationship between these storefronts, and it's pretty hard for participants to learn. So this is important because we're really taxing everyone's ability to varying degrees. And the key thing here is that some of our participants learn these environments under naturalistic visibility, so the buildings are opaque. But the other half of our participants learn the environments with this X-ray vision enabled. And then the interesting question is, how do we probe what they've learned and whether there's transfer of this knowledge to, quote unquote, a more naturalistic situation. So at test, what you can do is you take away X-ray vision from everybody. You say, here's your real environment. And we can probe their wayfinding knowledge in the or wayfinding performance in the environment. And this is sort of route bound in a way, even though participants may not be navigating a specific route that they learned previously. What happens is in wayfinding is when you go around a corner and the buildings are opaque, new cues unfold. And so you can sort of correct your heading and adjust your direction as the trial progresses. And we can kind of compare this with our pointing task in the environment, which is quite difficult. We we hide the rest of the environment from view on each trial, and you get one landmark, one storefront, and you're asked to point to where you think another storefront is in the environment. And there's no opportunity here to really correct yourself in this trial. So you take your shot, and that's the score you get. So this is a pretty challenging measure of their spatial knowledge. Okay, so to address our two hypotheses, then we're really interested in those objective measures of their spatial learning. And we're also interested in participants' sense of direction. And again, we're interested in that because maybe people who perceive that they're better navigators in their daily lives are in fact people who are better at using the cues that are available to them to traverse their environment. So we can ask whether some or perhaps all of our participants benefit from learning these environments with and without translucency manipulation during training. And we had two interesting observations here. One is that the high sense of direction participants in these controlled experimental settings with relatively tight availability of cues, they're actually not better navigators in general, but they do show an important benefit.
Thackery, you have 5 minutes left.
Okay, thank you. So if you compare the black bars, this is the opaque learning condition, you see the high sense of direction and low sense of direction people are not really performing any differently in the opaque environment training condition at test. But with translucency during training, there's a substantial benefit for having a high sense of direction. So these high sense of direction people in that top left plot actually outperform their own counterparts dramatically. We see the same effect with the pointing task. Participants with a high sense of direction aren't better pointers in general, but given extra cues, they really run away with it. So one way to summarize the results of this is to begin fleshing out a conceptual model that incorporates these individual differences and these environmental factors. So in this case, your difficulty in spatial relationship integration can be derived in part from your difficulty observing spatial relationships, and that's an environmental feature, and your ability, or perhaps your propensity, to integrate spatial relationships that you observe over the course of encoding. And we've gone on to replicate this work in larger scale environments. You see this recent example in the top right. We've looked at a situation where now there is a global landmark that participants can use to orient in the environment. We've manipulated the abstractness or sort of visual complexity. And I don't have time to go into this specific experiment today, but it was recently published and I wanted to make a quick plug for it, which is that if you return to this question, this research question here, when people encode the layout of their environment from a long circuitous route, a major cognitive antecedent of good spatial inference may be their working memory capacity. And that's because working memory allows us to sequence the spatial transitions that we tried out when we were exploring and the landmarks that they sort of revealed or led us to integrate that information into our map. So in this study that I will gloss over for you, we tested a model where working memory capacity contributes to this larger scale configural knowledge. And we were particularly interested in whether there was a curvilinear relationship between working memory capacity and spatial learning, here mediated by this difficulty in spatial relationship integration. The idea is that in theory, the difficulty of spatial relationship integration is going to modulate how quickly you saturate your working memory as you're proceeding through the environment. Perhaps it would also modulate whether you choose to engage in that sort of effortful encoding process at all. What's summarized on the right, just as a teaser for you, is clear evidence that this is the case. So this is plotted as an interaction effect. What you see is that higher spatial working memory capacity basically has no benefit on large-scale map learning when VR manipulations make spatial integration either really difficult or really easy. But when your participant traits and your environmental traits combine to create kind of a moderately challenging scenario, there's a big benefit on the right side of the plot for spatial learning in terms of your working memory capacity. Okay, so to get back on track though, what we can do to sort of synthesize the results from those two experiments is to say, well, these VR barrier manipulations really do seem to have potential for improving cognitive mapping. But their efficacy really depends critically on individual differences. And as we explore those individual differences, perhaps we can improve the sort of target of our manipulations. So one factor here in this case is the way that your perceptual barriers interact with your sense of direction. So why do barriers distort cognitive map attributes? Why does this translucency intervention actually work, at least in some of our participants? Well, the other research thread we've been following to try and get a handle on this is following up on some key neuroscience work, which you really don't need much introduction to, but briefly, the landmark discovery of grid cells in the rodent entorhinal cortex has suggested that these signals may help path integration among other processes in our sort of tasks. And the key finding I want to highlight for you is that Dori Derdikman from the Mosher group found that barriers in a hairpin maze environment, as shown on the right here, fragment grid cell maps into multiple linearized submaps of the space. So with the advent of computational methods for modeling grid cell-like signals in fMRI, this seemed like a great place for us to start in trying to understand the neural mechanisms that underlie the disruptive effects of barriers in people. So what we did was we generated a virtual hairpin maze environment. You can see examples in the top, the left, and the center here where the way this worked, participants followed waypoints as sort of foraging tasks. They follow a sequence of waypoints through the environment. And they would follow these waypoints through open fields, example in the top left, and a hairpin maze, example in the middle. And the beauty of this waypoint-guided design is that we can perfectly match behavior between participants and between experimental conditions. So here we can look at how barriers affect grid-like signals in human entorhinal cortex while factoring out the possibility of behavioral differences between individuals navigating those environments. So in the interest of time, I'll skip right to the data and some of the bullet points. So what you see in the top left are some really nice hexadirectional signals in human entorhinal cortex in both indoor and outdoor open field environments. And what's critical is that when we...
Thackery, sorry, only 2
Yes.
minutes left with
Okay, thank you.
questions. Okay.
Yes. Next slide, actually. So what's critical here is that when we introduce the barriers to the environment, this significantly disrupts these grid-like signals. And you can see this in the radio plot in the top right, as well as in this visualization where we look at different candidate periodicities in the data. And again, the key thing here is that the behavior is matched between these environments. They're navigating the same route through what's otherwise the same space. Now, we can't say whether this result is really related to the same phenomenon that the Dori observed in rodents, but I did want to highlight that we also saw this remarkable restructuring in the way that entorhinal cortex responded during this task when we introduced barriers. So we discovered that we get this fourfold rotational periodicity where these entorhinal signals align not with participants' navigation per se, but with the barrier structure of the environment. So taken together, this study suggests that one reason barriers may disrupt our cognition in these tasks may be by fragmenting metric information that putatively comes from entorhinal cortex. So to sort of conclude here, the direction we're going now is to explicitly make that link. So how do the effects that we see in entorhinal cortex of these barriers relate to the cognitive differences that we can tie to affordances and perception in our cognitive tasks I showed you before? And one direction we're going to go here is to zoom out a bit and look at broader scale functional interactions between entorhinal cortex and the rest of the navigation circuit. And look at this in the context of people learning broader scale environmental structures, such as this chaotic layout of old Atlanta here. So I'll end with a thanks for your attention. My collaborators on this work here are highlighted in yellow. And I also just wanted to say, if you find this work interesting, I find your work interesting too. So please don't hesitate to reach out. I'd love to think of other collaborations along the way. So any questions?
Thank you so much, Thackery. So yes, there are questions in the question and answers. The first question from Anis Paradis. Do you think barriers have different effects in different species or with aging, depending on whether the species or individuals rely more or less on distal cues and or visual exploration?
That's a great question, and it's very prescient, I guess. The shortest answer is yes, I think it does, and I just submitted an application to try and look at this in aging, because we know, for example, that people's perception of self-motion changes from visual cues as they age, and barriers affect optic flow, for example. Optic flow gets anchored to barriers. So there's a really interesting direction there to look at, not just between species, but even within humans across the lifespan, at how barriers may differentially affect our spatial learning.
Yep, absolutely. A second question from Marie Egarty. What are the applications of this research? We can make barriers transparent in a virtual world, but they are opaque in the real world. Can this research give us insight into how to train navigation ability or low SOD and low working memory individuals?
That's a great question. So, we haven't tested that directly, but I did make a big deal in the beginning about this transfer point. And so one thing to highlight from both those first two cognitive experiments is that we actually train people with translucency and or penetrability, but our tests of them in both studies are in opaque, impenetrable environments. And so at least in this still synthetic virtual setting, we can say that any benefits you got from training actually sort of hold up or perseverate at test when you have naturalistic visibility and affordances. So the hope is that you could, if you had a good simulation of a real environment, maybe you could engender better knowledge in, say, ambulance drivers and emergency responders. But we don't know. We have to test this.
Yeah, sure. Okay, well, thank you so much for this talk and for your answer to this question. We have to move on because of the time. But anybody who has more questions, you can email Thackery.
Yes, thank you.
Bye, see you. Okay, so we are going to welcome our next speaker, who is Matteo Carandini. Are you on stage, Matteo? Not yet.
Yes, can you hear me?
Yes, but I cannot see you. Is it on purpose?
No, let's see.
I can see your presentation, at least.
I can see you. I can see the presentation. Am I doing something wrong?
No, it's just we are not seeing you, but it's okay if you do.
My camera is on. Oh, they can see me. Michael Hassamo can see me. I'm sorry okay. You can't. But we've seen each other recently.
Okay, so let's go. We see you. Okay, let's go then. So again, welcome, Matteo. So Matteo Carandini is from UCL, and his talk today will be about studying the interface between vision and navigation and back. So go ahead, please.
Thank you for having me. Can you see my mouse when I move it around?
Yes, we can.
Okay, fantastic. So in case my internet dies, I wanted to say two things right away. The first is that this is a fantastic meeting. I apologize for my many comments. I promise I'll rein it in. But it's also the fault of other people who are sitting in the back. Somebody called Eleanor, Roddy. These people. Anyway, and then the other thing I wanted to say. The other thing is thank you for inviting me. And this navigation community seems to be way nicer than the vision community. So I'm hopefully here to stay, and hopefully you'll keep on welcoming me. In case my internet dies, the message of this talk is that we started research trying to understand how we go from vision to navigation. And then we ended up studying the opposite, how navigation influences vision. So we've heard, even in this meeting, how important vision is in guiding navigation. And we even heard in the last talk, well, a few talks ago, that bats, with all their echolocation and all that, the number one thing they use is vision to decide where to go. And yesterday, Mariam Najafian Jazi, if I'm pronouncing it right, gave a beautiful talk, a mini talk, showing that she was training these mice to go to a certain place, and then they had to head back to a port, and they were very good at it. When the lights were on. And of course, when lights are on, we know that the navigational system in the brain, for example, grid cells, look beautiful. But when lights are off, all hell breaks loose. I mean, these mice were terrible at navigation because probably they were turned around when they were looking for that blue thing. And grid cells, at least in mice, seem to go away. Now, you guys are the experts. Maybe not everybody agrees that grid cells disappear in darkness. I know this study, and I really like it. But I just want to say, light seems to be very important. And in fact, if you were to be blindfolded, the number one thing you'd worry about is how would you navigate? Except we're all stuck home, so it's pretty easy to navigate anyway. All right. So there has been increasing evidence that navigation might actually influence vision. And so, for example, there were these two papers. This one came out in 2015. This one came out in 2016. I'll start with the 2016 one. It was a study in virtual reality. Virtual reality obviously has changed a lot of what we do. And there were mice running in a virtual reality where first they saw pattern A, then pattern B, then pattern A, then pattern B, and then other stuff. It got complicated after that. And these authors, the Keller group, were able to, by imaging the activity of lots of neurons in primary visual cortex, to decode whether the mouse was at the first A or at the second A, even though visually they're similar. They're not identical because there's other stuff in front of the animal, but they're similar. And the group of Haggerty and G, they showed that place cells sometimes fire more, and when they do, it's on the same trials as V1 fires more, and place cells sometimes displaced to the left or to the right in a track. This is in real reality in rats, and also V1 responses appear to be displaced in the same trials by the same amount. So to study these phenomena, my collaborators, Aman Salim, Mika Diamanti, and Julien Fournier. Julien is very well known to Laura. I think they're in the same building. So Aman has a lab at UCL, Mika is a postdoc at Princeton, and Julien is in Paris. We started a series of studies in which mice would navigate virtual reality, and they would, crucially, they would go through a corridor where the patterns repeat identically. So in the first half of the corridor, the same things happen, first a grating, then a plaid, and then in the second half, first a grating and then a plaid. And this is crucial because it means that we can create situations where things look the same, but you are in two different places. And I understand that this is pretty standard. There was a talk yesterday about human virtual reality that had similar thoughts. But so now, if a neuron were purely visual, it should respond the same way in the first half and in the second half. Of course, there could be issues of running faster in one and slower in the other, but let's ignore these. These are all taken care of in the papers. And so what we found is that instead, some neurons in primary vision, so what's happening is here we are imaging with 2-photon imaging sections of primary visual cortex, the parts that are looking to the side of the animal. And we're imaging a bunch of, hundreds and hundreds of neurons at a time. And we can ask, when are they active? Now, the activity, of course, is fairly slow because this is GCAM6. But you can see that some neurons responded twice the same way, the first half of the corridor, the second half of the corridor. But other neurons, which is what you would expect from a perfectly visual neuron, but other neurons responded more in the first half or more in the second half. Now, these are not place fields, okay? These neurons are responding because there is something on the screen that is driving them. This is visual cortex. But crucially, if they were purely visual, they would respond identically in the two halves, and many neurons don't. If you now go plot a snake, this is what we call them in the lab, of all the neurons we recorded, you see that most neurons respond more strongly in one place instead of responding twice, okay? But this is a snake that was created by aligning neurons by their peak. And when you do this kind of thing, you will always find a snake, even when there isn't one. So be very thoughtful of that when you review papers. So what you need to do is use half of your data to find the snake, and then use the other half with the same ordering of neurons and see if the snake is still there. And the snake is still there, which means that spatial modulation in primary visual cortex is not only there, but it's also robust. Now, how strong is it? Even in these other half, which were not used to align the data, you can see that there's a central peak. Each neuron fires once in the corridor, and the second firing is much weaker. How strong is it? Well, we can measure it. Here are all of our neurons, thousands and thousands and thousands of neurons recorded with lots of, not only looking to the side of the animal, but also in front of the animal, etc. And we can compute a spatial modulation index, which is the kind of thing that you would define if you wanted to calculate spatial modulation. It's a very easy thing. Basically, it's 1 if you have a single peak, 0 if you have 2 identical peaks, meaning purely visual, and -1 if you're in the domain of noise. Like in half of the data, you decided that one peak was bigger, but in the other half of the data, it's the other peak that is bigger. Then that means negative, okay? And clearly for V1, we're very far from 0, meaning this is the 50th percentile, the 25th percentile, the 75th percentile. You can see that they're way above the strong spatial modulation indices. But now you could ask, could this be coming from the thalamus, the visual thalamus? The LGN is the nucleus of the thalamus that gives input to V1. The answer is no. We imaged the inputs coming from LGN into layer 4. These are synaptic boutons, and this is what they do. They respond twice, which is exactly what you would expect for a purely visual neuron. And so in fact, this is what we find in LGN boutons and in LGN neurons, which we also record with electrophysiology. And then another thing you may ask is, wait, how the mouse can't be born with this preference for modulation for spatial position in V1. It needs to know about this environment, like the kind of thing you guys would say about CA1 place fields. And indeed, this selectivity grows with time. This is the first day the mouse is in that corridor. We're back at V1, by the way. Third day, day 5. These are not exactly the same neurons. We didn't make an effort to match them, but you can see the selectivity, spatial modulation grows. Now, we can ask, does this spatial modulation get stronger as you leave V1? And for example, as you go into parietal cortex, where we've long known that there were spatial signals? The answer is no. We find pretty much the same amount of spatial modulation, if anything, a tad less, but this is in the noise. So it's as strong in V1 as it's going to be in cortex. And so the summary up to here is that neurons in visual cortex are modulated by spatial position. This spatial modulation is not inherited from the visual thalamus, so it's established in cortex. It arises with experience, and it is fully established already at the level of primary visual cortex. So next, we asked whether these signals of spatial position are actually consistent with position signals that are, of course, in CA1. And so to do that, we record at the same time in V1 and in CA1. And so we got lots of neurons. In this case, actually, the snakes are not cross-validated because I already convinced you that they exist. And so, of course, we can decode where the animal is based on what V1 and CA1 say. Of course, that's obvious. And this is the kind of stuff that you guys in your field do all the time. But now, because we're recording from them at the same time, we can ask whether when CA1 makes a mistake, does V1 make a similar mistake? So if CA1 on a particular moment, on a particular trial, says that the animal is, say, 3 centimeters ahead of where it actually is, does V1 make the same mistake? This is this kind of trick that we use all the time when we study decision-making, which is all about studying mistakes. And so now, this is not visible here because these are averages over lots of trials. And so, but when we do calculate what is the probability of an error in V1 given the probability of an error in CA1 in decoding, we find that they're fantastically correlated. And so in those cases, which are not common, I mean, usually CA1 and V1 are great. They don't make mistakes. But when CA1 makes a mistake by, say, 5 centimeters, V1 makes a mistake by 5 centimeters in the same direction. Okay?
Matteo, 5 minutes.
Thank you. So we find that this spatial modulation is not inherited from LGN, and it arises with experience. It is fully established in the cortex, and it correlates with the spatial position encoded in hippocampus. Now I'm going to ask a question that for you, you probably think you already know the answer, which is when V1 and hippocampus say that the animal is in a particular place, is that where the animal physically is or where the animal thinks it is? And because you probably like to describe yourselves as cognitive neuroscientists, you probably think, of course, that hippocampus is the mind of the mouse. That's where the mice thinks it is, if the hippocampus says so. But how can we test that? And I know John O'Keefe a while ago had an experiment that basically got at this. In this kind of environment, we can ask where the animal thinks it is by asking the animal to do a performance, and the performance, the task is to lick in a particular region, which is shown here in green, but the mouse can't see it. Okay, so the mouse needs to learn that if it licks by sticking out its tongue in this part of the corridor, it will receive a reward. By the way, this is how all the experiments up to now were run. All right, but I didn't need to tell you that. And now the good thing, which we did it as just to train the mice, but it turned out to be a great idea, is that we allow mice 5 licks. In other words, it's okay to lick 4 times in the early zone, as long as then you lick in the correct zone, you will get a reward. But if you lick 5 times in the early zone, that's it, you're out. And if you've licked in the correct zone, and it's fine, we will allow you to keep on licking in later, okay? So we can now define early licks and late licks. And we can ask what happened in CA1 and V1 when the mouse licked too early or licked too late. Are the signals consistent with CA1 and V1 telling the mouse, hey, you're already at the place where you should lick. Go ahead and lick even when the mouse is not there, right? And so that's exactly what happens. So, of course, the decoder works perfectly in the correct licks because that's how it was trained. So I'm only showing you now early trials and late trials. And this is the decoder from V1. This is the decoder from CA1. And hot means V1 thinks the mouse is in a particular place, or the decoder does. So let's just focus on V1 because it's identical to CA1, which is kind of the message of this talk. And as you can see, in early trials, the mouse licked too early. Okay, it licked before getting in the reward zone. Guess what V1 was saying? V1 was saying that the mouse was already near the reward zone, and vice versa for late trials. And if you go now look at where the mouse actually licked, the fraction of licks was very nicely peaked at the place where CA1 and V1 said that the animal was in the lick zone. So when CA1 and V1 say the animal is in the lick zone, the animal licks. So this, to me, is as close as I can get to the minds of the mouse and about what the mouse is thinking. So the spatial modulation that we're seeing in the visual cortex encodes the mouse subjective position, as does CA1 encoding, which maybe was obvious for you. Now, I don't have time to tell you much more. I just will summarize 3 results with just words. But this does appear in these papers. One of them is in bioRxiv, one in Current Biology, and they're recent. We find that these spatial signals are influenced by idiothetic cues, just like in CA1, meaning if we change the length that the mouse needs to run physically, as if it was harder to go through the corridor, that affects CA1 receptive place fields and V1 responses similarly, although vision wins by far relative to idiothetic cues, as you might have imagined. We also find that there's the position encode in CA1, in V1 oscillates with the theta measured in CA1, and we find that all of this requires active navigation. Otherwise, we don't really see the spatial modulation with passive viewing. Now, if you look at all these are all things that you've basically already heard about in papers that you've read about the navigational system, right? We're just repeating these findings in V1 and in higher areas of the visual cortex. So to summarize, I know that I'm at 15 minutes, so I'll end right now. I will summarize the results. Our results say that we find navigational signals all the way throughout visual cortex. And in fact, a paper by Cindy Poo in the Mainland Lab recently found them even in olfactory cortex. And as far as we can tell, these are the same signals that are encoded in CA1 and probably in other parts of the navigational system. And questions that we would like to address from now on is how do they actually get to visual cortex? Of course, each of you will have your favorite places by which they could get there. And do they only modulate responses? Remember, I'm not telling you that visual cortex has place fields. I'm telling you that the visual responses of visual cortex are modulated by position. But can these signals also maybe drive responses? And also the other question that I have is this just something that happens in mouse or maybe in rat, but not at all in primates like us? I started my career recording from the monkey primary visual cortex. The monkeys were anesthetized, so I have no idea what they would have done in navigation. But it was an incredibly beautiful system. I would be surprised, maybe I'm wrong, if it was modulated by position. But I think that our friends who are recording in marmosets are in the best position to answer this. Thank you so much for your attention, and I hope you guys are still there because I'm getting zero feedback, but I hope you enjoyed it. Oh, I can see Lo. Okay, hi.
Yeah, thank you so much, Matteo. It's a really fascinating work, and that, yeah, we knew a lot about already, but very new study. You showed us.
That's a backhanded compliment right there. All right.
Yeah. A lot of questions, so I tried to go through. So sorry for the pronunciation of your name, Shinme Purandare. If there were catch trial when the expected A was skipped, would the V1 neuron still fire?
That's a very, yeah.
Basically, would the expectation based on position override the stimulus presented?
Well, that's a really good question, and it would speak to whether some part of the brain actually has an expectation of the stimulus being there. We didn't do the experiment. It's the kind of experiment that Georg Keller does. Probably our labs disagree on where these signals come from. In my opinion, they probably come from the hippocampus and places like that. And in their view, they come from the prefrontal cortex. We didn't do the experiment, so I can't answer. I think Aman Salim is doing this kind of stuff in his lab.
Okay, thanks. A second question from Arne Ekstrom. Could you speculate on whether the CA1-V1 similarities are due to distributed redundant codes, or that V1 and CA1 are performing unique and separable computation related to navigation?
So I think V1 is doing its job, okay, and it's doing vision, and we know that it's also doing other things. For example, you can find signals related to body movement, like in terms of like muscle action, etc. But luckily, we've discovered that these signals are orthogonal to the visual signals. So I suspect that also these navigational signals are probably going to be orthogonal. So if somebody wants to decode them, they will be able to, but if somebody wants to ignore them, they'll be able to as well. And so, but in terms of whether they are redundant, these codes between V1 and CA1, I suspect that it's the same signals. I think the navigational system is getting its estimate of position mostly from vision. Come on, let's accept that. Then if the light is off, we'll use some other methods. We'll go by the wall and touch it with our beard and whiskers, sorry. And otherwise, it computes an estimate of position and broadcasts it very widely to all over the brain in a mouse. In us, I don't know. I hope this answers it.
Yeah, okay. Yeah, I'm sure you did. Okay. Maybe one or two more questions.
Oh, silencing hippocampus. I can read the question.
So what is your guess? What happened if you either could silence V1 or hippocampus? What is the causal pathway?
Okay.
Would you get V1 position decoding without hippocampus? That's a great question. It's a super tough experiment, but luckily it was done in the lab, I think, of Vincent Bonin. And the result, and they were looking at these snakes in retrosplenial cortex, and the virtual reality, I think it was tactile and not visual, but you can find these snakes of encoding of spatial position also in retrosplenial cortex, and maybe that's actually how they get to V1, by the way. And they then did chemogenetics, I think, or something chemical to inactivate
large chunks of hippocampus, and the snakes in retrospinal cortex disappeared. That's my summary of their work. I might not be doing a good job there. I think it's a PNAS paper.
Okay. No, fair enough. Last question, because of the timing, from Eleanor Duvel. Do you know if the V1 Do you know if the V1 spatial learning effect is dependent on hippocampal replay?
I don't know because I've never measured hippocampal replay, but the first paper that made people suspect that the visual system and the navigational system were actually intimately connected was a paper by G and Wilson in 2007, where they found that replay or ripples, I can't remember if they were talking of ripples or replay because I think the two are related, were happening at the same time in hippocampus and V1. G and Wilson, Nature Neuro 2007. Thank you, Mayank. And so I suspect that all of this has to do with that. But I don't know. I haven't done that experiment.
Okay, well, thank you again, Matteo. I think we have to move on. And again, there is a lot of more questions that you can ask directly to Matteo. That's
thank you.
Not a problem. See you. Okay, so now we are welcoming our next speaker. Can you hear me?
Yes, I can hear you. Can you hear me and see me?
Yes, both.
Okay.
So everything is fine. So I'm very happy to welcome Adolfo Bronstein from Imperial College London. And I think you are going to talk about neurological control of vestibular navigation. That I have to say, I'm really looking forward to hear.
Okay.
So you can go try to share your screen and we'll see if it works.
I thought I was. Can you not see my screen now?
I can see you, but not your presentation.
Okay. Yesterday, I had to share the entire screen. I'll try that and see if that works.
Okay.
No?
Not yet.
I had that problem yesterday, I'm afraid.
Are you on Chrome?
Yes. As far as I know, I am on Chrome, really.
Okay. So you are trying to share it and it's not working, right?
Okay, let's see this. Any luck?
Okay, yeah, it's coming. Yep.
Okay.
That's perfect. You are on.
All right. Okay. Can you see me as well?
Yes, we can.
Okay, thank you. So this talk is going to be perhaps reverse to the preceding one because as vestibular scientists, we have to cancel out the role of visual input and often end up doing our experiments in the dark. But don't forget that we also have to navigate and orient ourselves in the dark. So it is a reasonable question to ask. How do we navigate with our vestibular system? In terms of vestibular perception, which you need to perceive the vestibular stimulus if you're going to use it for navigation, there is not a single vestibular perception. The vestibular system gives you information about the velocity of the head, usually measured in degrees per second, or it measures the displacement, not so much in linear coordinates, but in angular coordinates as well. And of course, also, you actually have the possibility of, through your vestibular system, sense the orientation of your head with respect to the gravity vector. So I'm going to start with a few examples from past work using the head displacement or odometer function of the vestibular system, which you could correlate or compare with the compass or the heading direction of instruments in the aeronautical field. Much of this work has used this experiment that my colleague Michael Gressy devised many years ago. We call it the self-rotation experiment. The subject sits in a rotating chair. The rotating chair is controlled by the computer, so you give a rotational stimulus, say 90 degrees to the right and in the dark and with no hearing cues. The subject brings himself or herself with the joystick back to the starting position. That would be a return to start, like a path reversal scenario. Can you see my cursor, please? Can you see my cursor?
Yes, no worry, we can see
so this
everything.
is the joystick that allows the subject to control the chair. So, and these are recordings of this experimental situation. The subject is sitting on the chair. We give them a displacement, a rotational stimulus in one direction, and the subject has two extreme possibilities of response, if you like. One is copying that response to return back to the starting position, or to navigate more spatially, and for this we actually limit the velocity available to the joystick, so the subject has to sense when he is back to the starting position. What we did in this early experiment with Barry Simongal was doing his PhD with us. He actually mounted the TMS coil on the rotating chair, so we could, by giving repetitive TMS to the posterior parietal cortex, hopefully disrupt the process of encoding of this spatial stimulus. And these are the responses during right posterior parietal cortex virtual lesion. We were able to disrupt rotations to the left, and the effect was more pronounced with right parietal lesions than with left parietal lesions. And we know that there is a predominance of the right-sided hemisphere for vestibulospatial function. This was later confirmed in another paper where patients with stroke were collected, and those patients with a stroke and spatial dysfunction during a similar type of task like the one I've shown you, the area of overlap of the lesion is in the temporoparietal junction. So both the TMS and the stroke data conclude that vestibular spatial perception is localized to the posterior parietal cortex and to the temporoparietal junction. What was interesting in this study is that motion perception, we was separately investigated, was preserved. So the perception of the velocity of self-motion was preserved, which indicates that velocity and spatial perception are dissociated in the brain. So talking about head velocity of the speedometer function of the vestibular system and the brain, I got interested many years ago when putting people in the fMRI scanner. We noticed that during self-motion sensations, the area that was particularly lighting up was this little area of the cerebellum, which is the nodulus. The nodulus is the central part of the vestibular cerebellum. So it did make perfect sense that that area was getting activated during the perception of self-motion. So for that reason is that then I went to study more formally in a group of patients with midline vestibulocerebellar disorder, what was their perception of rotational angular velocity. And for that, we devised this technique where the subject has a joystick, a wheel, and they turn this wheel very quickly if they feel that they are turning quickly. And as the sensation becomes less and less rapid, so they begin to slow down the rotation. And these are two examples of those recordings. You rotate the person at 90 degrees per second constantly. And as you can see, the normal subject has a prolonged response, which decays, as you know, if you keep rotating, the vestibular system loses the signal, whereas in the cerebellar patients, this drop out of the rotational sensation was much rapid. That's why we put it as a name to the paper, that simply these patients with cerebellar disorder had reduced velocity perception. just to give you a little bit of background, that was, I'm going to describe now our current work using the same type of patients with downbeat nystagmus, which is a syndrome that we know is restricted to the vestibulocerebellum, and the vestibulocerebellum is important in that it probably mediates one of the accesses of the vestibular system into the cortex and the hippocampus, which might be used for navigation. This work was done just before lockdown by Matteo from Athens, who managed to take the data away with her, and so this was done mostly, except the experiments, on Skype. So we're looking at these patients with the question in mind: if these are all the various pathways whereby the vestibular system can potentially reach the hippocampus and other relevant cortex, entorhinal cortex, and so on. And we were going to investigate in these experiments the vestibulocerebellar cortical pathway with a secondary question in mind, which is what might the signal in this pathway be? So our patients are going to have lesions in this area, and so we hope to be able to understand a little bit more. So for this paradigm, we use again this self-rotation experiment. We give a stimulus to the subject brings himself back to the starting position in two ways. So this is the stimulus out, and one instruction to the subject is, okay, return to the start, go back to your starting position. Initial stimulus, but then a second, more spatial, more centrally integrated function, which is, okay, we give you this stimulus, you have to find your way back to the starting position, going around the circle. So this one, we call it the CTC, or complete the circle condition. I don't know if you can see my willing subject here. I cannot see myself, so I hope you can see my subject here. So if this is a stimulus of 90 degrees now facing the camera, so the return to start, or RTS, the subject will go back to the same position. But if we give a CTC stimulus, the subject will go to this position, and we'll have to go around the block, so to speak, to find the starting position again. It's like going to a shop which is next door to you, round the block, rather than walking directly. And these are examples of the stimulus. So this is the velocity which we distorted deliberately so subjects could not copy that. And this is a return to start, RTS, so the subject goes in the opposite direction. And this is the displacement signal. So in this case, a 90-degree stimulus was only given a 70-degree response. So this was a hypometric response. So the gain of this response would be below 1. An optimal response would be 1. Another example now for the CTC, or complete the circle task, the velocity stimulus in this case. To the right, the subject will have to carry on rotating the chair to the right until they find the position. This was a 120-degree stimulus, and the subject correctly moved the chair in the same direction, 240 degrees, to complete the circle. So these are some of the scans of the patient. We tested five patients in this sort of 60s and 70s of age range. In this patient, age 60, you can see a small degree of midline cerebellar atrophy here. This is a 66-year-old patient. The cerebellar is a little bit more intact, but as I mentioned, we know from a lot of various different types of information that these patients have a vestibulocerebellar syndrome. What they have is basically a downbeat nystagmus. So the eyes do this, a very vestibular cerebellar nystagmus. So, and these are the results of that study. So these are the healthy groups, and these are the downbeat nystagmus or cerebellar patients, and this is the gain of the response. This is for the RTS task, and this is for the CTC task. And, you know, I'm going to spend very little time on this because, as you can see, the responses of the patient completely overlap with the normal range, so no problem with the RTS and no problem with the circle. So there
Adolfo,
is this,
Adolfo, 5 minutes left.
Oh, okay, thank you. So this means that downbeat nystagmus patients show no abnormalities in vestibular navigation. The vestibulocerebellum does not convey spatial orientation signals to the cortex. The vestibulocerebellar cortical pathway likely carries a velocity signal. This is supported by the clinical picture that the nystagmus they have is in fact a velocity bias in the vestibular system, and also by my previous finding of reduced velocity perception in these types of patients. So, and now to something completely different, which is the vestibular contribution to navigation in normal individuals with a normal vestibular system. And here we're going to assess the age effects, particularly to see if there is a uniform decay across different navigational tasks, and whether the exposure to map apps or GPS system affect your sense of direction. So a terrible slide. We tested 24 subjects, a number of vestibular conditions there, eliminating low frequency responses that will be difficult for the vestibular system to process. But we added subjective assessment. We quantified map app use of phone GPS from 1 to 10, 10 being I use it all the time, don't go anywhere without it, which is a real response by a real subject, and measure the Santa Barbara sense of direction questionnaire as well. So these are some examples of the Santa Barbara questionnaire. This could be a good one. I don't have a very good mental map of my environment. And these are some of the results of these experiments in young and old subjects as a function of the amplitude of the stimulus for the return to start. As you can see, there is a slight decline with amplitude, which is explained on the basis of the vestibular physiology, a little bit worse in the old. In the CTC or round the block task, this is a flat response, essentially getting worse for high amplitudes, which is the reversal of this. This is fine. This is all explained by the plain vestibular physiology. But there is no major difference between the young and the old, particularly in the central range. We did not confirm statistical difference there. But when we bring into this equation the subjective parameters, for instance, the map app use and the Santa Barbara sense of direction score, we find this perhaps expected relationship that the more people use their maps, the map apps in their phones, the worse their sense of orientation it was. So this is a chicken and egg, like any correlation at work, but we thought that if we throw in a third variable, the age of the subjects, which is plotted here in the x-axis, we can see, as expected again, that the use of the navigational maps from their phones is much, much less in the elderly. But at the same time, they seem to have a better sense of direction. So this is not quite significant, but you know, this explains a significant portion of the variance. So, because I have no reason to believe that biologically the young and the old are different, as how they were wired up in the first place, I would tend to assume that it is the map app use that led to a reduced sense of orientation in the Santa Barbara score. And when you bring in the other factors here, which are the vestibular navigation parameters, you can see that this complex complete the circle task, the going around the block task, is related, significantly related to the Santa Barbara score. So how do we bring all these parameters together, all these correlations together? Okay, one way is with factor analysis, and we actually put all the variables in, and this model explains 71% of the variance. We had basically two components. The first component group, the Santa Barbara sense of direction score, the age of the subject, and how much they use the map apps. The second parameter, or the second component, which explained 30% of the variance, also included the Santa Barbara score, but it did include both vestibular navigation tasks, the RTS, and more significantly so, the CTC, the round the block task. So this was indicating that If you actually look at the Santa Barbara score and throw all the variables into a multiple linear regression analysis, the one that remains significant is the CTC gain, the vestibular navigation complex task, which remains statistically significant. So this is my last conclusion slide. Vestibular navigation, preliminary conclusions. We've done really some of these slides today. Aging is associated with less use of map up, better sense of direction, a decline in path reproduction, but not in path completion. So the simple task gets worse in the elderly, but the complex task does not get worse in the elderly. And I wonder if this is a kind of central compensatory process, as when we lose our memory as we age, and I have plenty of experience on that, we begin to take more notes, you use diaries more efficiently, and so on, and you manage to, in theory, seem to have better memory than young people. So factors involved in developing a sense of direction include age. I'm sure this is up to a point. Map up use and vestibular navigation skills. So from this work, it seems that map up use inhibits our internal sense of direction. I thought that this was one of the first papers to show this, but yesterday I found that there was a paper in April this year in Scientific Reports showing something along these lines, I regret to say. So I think that this study is probably the first to show that pure vestibular navigation skills, remember all of this happens in the dark, influences the subjective sense of direction. So pure vestibular navigation skills do influence the subjective sense of direction. Thank you.
Yeah, thank you very much, Adolfo. So, yeah, there is a question from Jean-Laurence that actually goes with a kind of question I have in mind too. So he's saying the main function of the nodulus is to deal with rotation in vertical planes and translation. We can come back to that later. Could it be that the vestibular contribution to spatial orientation relies on vestibular nuclei projection or the flocculus?
Well, I wouldn't know what differences the nodulus or the flocculus will produce. The nodulus is a central structure, as you've seen in those scans. The flocculus is a bilateral structure, which is probably more lateralized. So lesions of the flocculus on one side clearly lead to lateralized problems to one side or the other, whereas the nodulus is more central, midline, more vertical. But in terms of the processing and in terms of the inputs, which is essentially vestibular, they probably do a very similar job. That's my gut feeling, but I have no data on that.
On the same line, and before going to the second question, you said that the vestibulocerebellum carries velocity signal, and you essentially talked about the rotational aspect. What would you predict in terms of translation with the
yes,
cerebellum?
Overall, there is much less work on the translational during the linear VOR than during the angular VOR. However, one of the things that we are absolutely sure as clinicians is that lesions in that part of the cerebellum have a profound, in fact, impact on the eye movements or the subjective sense of dizziness of the patient. For instance, if you have your subject like this and you look at the eye movements may be pretty steady, and then when they stand up, the eye movements become completely wild. So one of the things that happens with this area is that they seem to modulate and possibly modulate down the influence of the gravity vector. So you might actually, all of you can ask yourselves, how come that I have this massive acceleration on my head acting all the time? A Ferrari, you know, when it really accelerates approximately at 1/4 of a G. And yet, we are all walking with 1 G down our brain all the time, and we hardly feel it if we put the head like this or like that. So, but when you have lesions in this area, the system seems to get disinhibited, and passing from this position to this position produce massive symptoms in the patients and massive eye movement responses. So this part of the cerebellum may be actually controlling, modulating the gain of the vestibular responses.
Yep. Thank you. And last question, maybe from Annelise Paradis. Do you think there is an implicit versus an explicit sense of direction that could explain the differences between vestibular abilities and introspection results?
Pass. Sorry. Why don't you email me? And you probably know more than I do about this. Please email me with your comments and your views, and we'll have a nice conversation. Thank you.
It was not from me. Huh? It's
okay. Yes, from the author of
okay.
that question. Please email me. Thank you.
Okay, so Annelise, you can email Adolfo directly. Okay. Well, thank you again for this presentation, and yeah, see you. Have a good night. Bye. Hello.
Hello.
You see me, I see you. Okay. Can you talk to be sure that I can hear you?
Hello. Can you hear me?
Hello. Yes, I do.
That's great.
Okay. Perfect. So you can begin to share. Yeah, it's working. So we are welcoming our last speaker of this session. So Nikolai Axmacher from Bochum University. We are really happy to listen about your talk about grid representation and path integration in Alzheimer's disease risk carriers.
Yes, thank you very much as well. I'm very excited to be here, and very grateful to the organizers for organizing this exciting online meeting. It would only have been nicer if we would have been able to meet in this beautiful location in northern Italy, but maybe we can do that at a later point in time. So today I would like to share with you some of the results that we had acquired in the last years about the relationship between grid cells and grid representations and path integration and their possible relevance for understanding early Alzheimer's disease risk stages. So if you look at the brain of a patient with advanced Alzheimer's disease, you can understand that at least in the near future, there's absolutely no treatment imaginable that could improve cognitive dysfunction at this stage. However, it is well known that Alzheimer's disease starts much earlier than the clinical symptoms, probably decades earlier, and you can find various different changes in brain structure and function at much earlier disease stages. And therefore, two key goals in Alzheimer's disease research are, on the one hand, to develop biomarkers to identify patients at early disease stages who may still benefit from treatment, and also to understand the early disease stages in terms of their neural level and also in terms of very early cognitive alterations. And one gene, or the gene, which is arguably the most relevant for these early disease stages, is apolipoprotein E, where the risk allele is relatively common, occurs at about 15% of the population. The risk allele is called ApoE4 and increases the risk for developing late onset Alzheimer's disease by a factor of 3 to 4. So the entorhinal cortex is among the very first regions that show Alzheimer's disease-related neurodegeneration, and there's evidence for very early neuropathology in ApoE4 carriers, and early means starting in the adolescence or maybe even childhood, so decades before any potential symptom onset. At the same time, as you well know, the entorhinal cortex contains grid cells, a fundamental building block of navigation, that are characterized by 6 preferred directions where the activity is higher as compared to the directions in between. Different grid cells along the longitudinal axis of the medial entorhinal cortex differ with respect to the spacing between their firing fields, but despite this variability, they seem to significantly share their preferred directions. And therefore, there's now evidence for a macroscopic or network marker of grid cell activity that is typically termed grid representations and was first described in a Nature paper by Christian Döller at that time in the lab of Neil Burgess at UCL London. So in the first study, we tested whether grid representations are altered in Alzheimer's disease risk carriers. So we recruited young students who were ApoE4 carriers and compared them to non-carriers and asked them to conduct a spatial learning task in the scanner, and then extracted their grid representations and found that indeed the risk carriers had a strong, substantial impairment in their grid representations in the entorhinal cortex. So notably, the participants here were really young. So the average age was about 22 years. Even though we found that the strength of the grid representation in a given individual was related to behavioral performance, overall, the risk group did not perform worse than the control group. How is this possible? So we argued that the participants with reduced grid representations possibly recruited compensatory mechanisms to make up for the impairment of their grid cell system. And indeed, we found that participants with reduced grid representations showed overall higher levels of hippocampal activity. Furthermore, we found that the risk carriers showed alterations in their navigational behavior. They navigated more closely to the border of the virtual arena, so in the periphery than the control participants. So there's a number of follow-up questions that we've since then tried to address. The first is, what is the electrophysiological basis of grid representations? This question is not so simple to address because obviously we would want to measure electrophysiological activity in humans in the entorhinal cortex, which is difficult or even impossible to selectively assess via non-invasive measures. So we conducted a task in epilepsy patients with intracranial EEG recordings. So these are patients who have a pharmacorefractory epilepsy and for the planning of the operation have electrodes implanted in various areas of the brain, including hippocampus and entorhinal cortex. And we tested for different possible electrophysiological mechanisms of grid representations. On the one hand, we tested whether they relied on theta oscillations, which are the predominant hippocampal oscillations in rodents and other species. Alternatively, we argued that maybe delta oscillations between 1 and 3 hertz could play an important role because it has been suggested that they are actually the human correlate of rodent theta oscillations. And finally, we tested whether broadband gamma activity as a measure of overall neural activity showed the pattern of grid representations. So we conducted the same task in patients with entorhinal cortex electrodes, had 9 patients in total, and found clear evidence for grid representations in the 4 to 8 hertz theta frequency range. We also found that these patterns were more pronounced when the participants navigated again close to the borders, suggesting that the grid representations were reset via the encounters with these visual cues. At the same time, research from Josh Jacobs' lab from Columbia University showed exactly the same findings. So during a different spatial navigation task, they also found in the entorhinal cortex grid representations between 5 and 8 hertz, which is a beautiful convergence of results here. So we are currently, so we believe that these findings are actually a first step towards understanding the link between grid cells and fMRI grid representations by showing how grid representations can be found in network oscillations at an electrophysiological level. And much more work remains to be done to link the activity of single grid cells to the grid representations and then eventually to fMRI. And if you're interested in this, we try to differentiate different scenarios, how the activity of single grid cells could give rise to network level grid representations. I'm happy to come back to that in questions, or you take a look in our recent article. So the second question we wanted to address is which specific navigational mechanism is actually supported by grid cells? So, of course, navigation in the real world does not rely on a single brain area or navigational strategy, but could rely on multiple different strategies, such as path integration, navigation related to allocentric cues, landmark-related navigation, and so on. And only some of these strategies are likely to require grid cells. And this may actually then explain why several previous studies, including our own, did not find behavioral deficits in ApoE4 carriers, despite some impairment of one of these systems in the entorhinal cortex. So in a follow-up study, we aim to develop a paradigm that allowed us to unmask behavioral deficits in ApoE4 carriers when they could only rely on path integration and not on allocentric spatial cues. And we also wanted to clarify the relevance of grid representations for path integration, similar to what has already been done in a previous study from the Wolbers lab in Magdeburg. And finally, track neurobehavioral markers of compensatory strategies. So we conducted a path integration task, the so-called apple game, in which participants first had to move towards an empty basket and then go to a variable number of trees until they found a tree with an apple. And once they had found this, they needed to go back to the original basket and remember the position of the basket. So importantly, there were different subtasks here. In one of the subtasks, the participants could only see the visual flow and use this for orientation in the arena. In another subtask, they could also use a boundary. And in the third subtask, they could make use of the information from a local landmark. We collected a relatively large number of participants across various different sites in Europe, in the Apple Game Consortium, and also conducted an fMRI study with a very similar paradigm in non-genotype participants. We are currently planning an fMRI study with the same paradigm in genotype participants as well. So what did we see? We found that indeed the risk carriers were impaired selectively in the pure path integration version of the paradigm, so that when they only needed to rely on the visual flow, but they were not impaired when they could use the boundary or the landmark for navigation. In that case, they were doing as well as controls.
5 minutes left.
Thank you. We also found grid representations in the posterior medial part of the entorhinal cortex, and found that the grid representations predicted performance selectively in the pure path integration subtask. So participants with more pronounced grid representations did better in the task, but this relationship occurred only in the subtask where they could only rely on the visual cue, on the visual flow. We next tested for neurobehavioral markers of compensatory strategies and found that the landmark information allowed the risk carriers to compensate for deficits. And so we found that all participants in the landmark task did better when the basket here, so the goal, was closer to the landmark, and were deteriorated in those trials where the basket was further away. However, we found that this relationship was more pronounced in the risk carriers. We also found that the risk carriers moved closer to the landmark. So when we tested the proximity of their navigation movement, we found that the paths were more closely related to the landmark than they were in the control participants. And in the fMRI version of the task, where, as I mentioned, we didn't genotype the participants, we found that the landmark-supported path integration showed a more pronounced activation in areas including the retrosplenial cortex. So to summarize, we have some evidence that ApoE impairs the performance of the grid cell system in entorhinal cortex, which physiologically relies on theta oscillations. We are currently planning to study how this system directly then relates to the activity of single grid cells in humans. As a result, of the impairment of the grid cell system in ApoE4 carriers, we found that they showed a reduced performance in pure path integration tasks, but whenever they could use additional spatial cues, such as landmarks or boundaries, they could compensate for this reduced grid cell system and do as well as controls. We also found, and I showed that in the first study, that this compensatory recruitment of these areas is related to hyperactivity, and there's some evidence that a sustained hyperactivity across many years, across the lifetime, may then yield to amyloid deposition, so actually may be causally relevant for the further development of Alzheimer's pathology and then to the spread of neurodegeneration from the entorhinal cortex to other areas in the brain. So, this is Anne Bierbrauer here, who did the Apple game study together with other colleagues. This is my group in Bochum, if you would like to join. If you're a prospective PhD student, please feel free to apply. There's a position available in a new project on grid representations. So thanks for your attention. I'm looking forward to questions.
Thank you very much, Nikolai. Very interesting. A lot of questions, actually. So the first question is from Paul Duchenko. Does your finding of decreased grid signal in 22 years old Hapoher individuals suggest very early pathology in the entorhinal cortex?
Yeah, that's a great question. And of course, I would love to give a clear answer. So I don't know is the short answer. But at least at a neuropathological level, studies from Brack and colleagues suggest that there is some hyperphosphorylated tau even in adolescence, and this is more pronounced in ApoE4 carriers. So there are some there's at least some evidence from neurodegeneration, even in these young participants. Whether this is really related to Alzheimer's disease is still not exactly clear. So there's I believe that in the future, tau PET and amyloid PET will be crucial to answer these questions about early pathology and giving information about the regional distribution of these changes.
Yeah, especially if I can add something. Reading a different paper on this topic, ApoE4 carrier, it looks to me that sometimes memory is not affected, whereas you have other type of behavior such as flexibility or reversal learning, or now you show beautifully path integration are much more affected in. So yeah, seems to be not hippocampal dependent, but something else for sure.
Yes, so the literature on ApoE4 related cognitive impairments in young participants is a little bit messy. So there's a recent meta-analysis suggesting that there's no overall cognitive impairment. I would suggest that it really depends on identifying tasks that rely on very specific brain areas. So for example, path integration in a condition that specifically relies on entorhinal cortex.
Yeah, for sure. Behavior matters, for sure. Okay, next question from Thomas Wolbers. How do you know that the past integration deficits stem from pathology within entorhinal cortex that compromises positional estimate? Could the deficit instead result from imprecise motion perception, which should also affect grid cell computation? Yeah, definitely.
Yes. It's a very good question, and I mean, I think it would also be very important to see whether similar effects can be found in real reality. So outside of the virtual environment, when it comes to the perception of proprioceptive vestibular information. So what we are currently planning is actually to conduct a whole battery of spatial navigation tasks, mainly on a behavioral level, in ApoE genotype participants, and see how they relate, and whether there could be a common trait marker, like stronger reliance on landmarks or boundaries on spatial cues, and this may be found in different paradigms.
Yeah, and Barry Simongal, okay, too late. The question disappeared. Okay. Next question, so from Nora Newcomb, you mentioned differences in ApoE4 carrier in childhood. Is that surmise or is there data?
I mean, the data that I was referring to is neuropathological data. So from children who passed away for reasons obviously unrelated to Alzheimer's disease, and whose brains were then dissected. And this is the evidence I know of. I'm not aware of any ApoE4 studies on navigation in children. I mean, maybe it's my ignorance, but then I would be glad to learn about this, but I don't know of any such studies.
Yeah. Question from Tim McNamara. Has anyone investigated ApoE4 carry path integration performance when they have body-based cues to self-motion? Fully immersive VR, for example.
Yeah, I'm not aware of this. Yeah, it's a good question.
A lot of work to do for you then. Still. Okay, maybe a last question. So let's see which one has the more votes, because there are still a lot of questions. So from Gregory Peters Bronstein, grid representation via fMRI were found in region outside the ERC. With regard to this, ERC considers the earliest close-up of Alzheimer pathology. Would you say grid representation in this region, DMN region, for example, are most resilient to Alzheimer pathology? Could they partially explain preserved performance?
Yeah, it's another great question. So, I mean, I think it is established in fMRI studies that these grid representations are not exclusive to the entorhinal cortex. Whether this is really, like, what is the electrophysiological basis for that, I think is much less well known. So, I mean, we plan in the future to conduct single unit recordings in human epilepsy patients, and then hopefully we'll get a better understanding of these grid representations outside of the entorhinal cortex. So whether, I think currently it's not even clear that in all cases they are similarly related to spatial navigation. They were also found in during conceptual navigation and so on. With regard to ApoE, I would assume that the effects should be more pronounced really for the entorhinal grid representations as compared to, for example, grid representations in the ventromedial prefrontal cortex, because of the earlier onset of neurodegeneration in the entorhinal cortex than in other areas. But that's a speculation.
Okay, well, I think we can stop here. I really want to thank you again for this great talk and the great discussion that followed. There was much more questions like in the previous talk. So again, you can contact Nikolai directly to continue the discussion. So we now have the session 6 that is ended. So don't go away. Please stay connected because we have still an exciting session, a Blitz Talk 1, that will begin at 9 p.m. CEST time, so Paris time. So see you there in 25 minutes. Bye.